So I'm just going to say a quick word before while the slides are coming up. So the talk I'm going to give is about the problem that I think we have more generally in science and in neuroscience these days, which is basically how much of in the literature we get results that we can't really trust, and what can we do to sort of correct for that? What are the causes of that irreproducibility? And it's been a problem that has sort of emerged more and more, and it's a real question because it really undermines the public trust in the scientific activity, and also obviously generally politically the funding that is going into the field. And imaging genetics has been one of the places where we can do better, and neuroimaging in general is one of the places we can do better with reproducibility. So we thought that is one of those things that we need to address. Yeah. So if you have a can you try F5 or F11 or Control L? No. This is just a PDF. This is there's no. It's just a. View, can you go in view maybe? If you try view and then and then full screen. Yeah, thank you. And let me try to move. Okay. Yeah, cool. Okay. Okay, so skipping the introduction. So reproducibility obviously is one of the major things. If we get a result in the literature, we want that to be something solid we can account and we can be sure of, or as much as we can. So I'm going to give a couple of evidence of the problem of reproducibility, quick word on the causes, and what should we do. So in general, I mean, I don't know if you probably have seen some of that. There was a nice editorial in Nature recently, actually a couple of years ago, showing all the problems of reproducibility. So the easy misinterpreted artifacts, the fact that big journals always try to get very unlikely but very sexy results, and therefore maybe more prone to have irreproducible results. There's a couple of good papers on the problem of what are our standards for statistical analysis. In epidemiology, I would really recommend to read the Ioannidis papers. The most famous one is the 2005 paper, telling us why most of the results that we see in the literature are probably false. In social science, it's been a plague, and people are starting, projects that are saying, okay, we are going to take the 30 most important results of the last 5 years and try to replicate them and see how many those are replicated. And that's the reproducibility project. There's Siemens et al. I think it would be an important paper to read as well, showing you how if you have a flexible way of analyzing data, you are increasing your risk of error very quickly and and dramatically, so drastically. So that's a very important paper to have in mind, that when you're analyzing data, if you have a flexible, you're not too sure exactly what you're going to do and you're trying different things, that's a very easy way to increase your risk of error. And in cognitive neuroscience, there's a good special issue on reliability. Oncology research, that's a very scary paper showing you that out of 50 major results published in Cell, Nature, Science, all those big journals, only a very few handful of them were actually replicated when a company tried to replicate those results. And in brain imaging, there's a couple of already papers out showing you that a lot of the brain imaging results are not that trustful. It's hard to know which ones are good. And in genetics, of course, we've seen the problems that was just referred to, the aspect that we have the multiple testing and a comparison issue, and again Ioannidis in 2007 showing you that 16 of the hypothesized SNPs that were thought to have an impact were checked and in cancer control studies, and results were largely null. There's many reference and warnings. The one from drinking for the fire hose is a good one as well. So what about imaging genetics? So first of all, why do we have a problem? One aspect of it is that things are getting complex, and the data description, size, computation, all those statistical methods are getting complex, and that's one aspect. It's harder to do, and we need to be more careful. One key thing, I think, which is probably more and more recognized now, is that because of the publication pressure, people tend to sort of go quickly and not do all the checks and the thing that needs to be done to make sure that the results are correct. Mistakes are easy to make, very easy to make. I mean, when I'm working with students and postdocs, and they come with their data, and I say, "Oh, have you checked that?" And "Oh, I've seen that." And that's so easy to make. And there's a couple of examples that the right versus left issue in brain imaging. There are very famous examples that are well known, and there are many more examples that are not that well known at all. It's just very hard. It takes time to check things. But the thing I want to concentrate on today is the power issue. And that was mainly Ioannidis' sort of a message in this paper. But there's a more recent paper by Button et al., the power failure in neuroscience, that I will relate to. So I will remind you what is power, what are the issues of low-powered studies, and a couple of words on the tools to compute power, and the question of what is our effect size. So what is power? First of all, the effect. If you have two populations, the means of two populations, the effect would be like the difference of the means of those two populations. What's the standardized effect, which is often referred to as Cohen's d, which is just this difference of mean divided by the standard deviation of the data, not of the mean, the standard deviation of the data. So it hasn't got the size of the group in that here. And the z, or the statistic that you're going to get, and from your p, and then eventually go to that statistic to the p-value, it's basically the same thing but divided by the square root of n, or something close to the square root of n, which n is the effect size, the number of subjects in your study. So that's a quick recall. And so what is power? Well, let's say we are in the framework of hypothesis testing. So we have a null hypothesis. We think that the two means are equal, and that the difference is 0. And this is our distribution of the sampling distribution of this difference of mean. And to get the power, you need to have what is the alternative distribution. So you need to know what is your expected effect size, what kind of difference of means, if they are not 0, is going to happen. And then you cut the null hypothesis distribution here at 5%, and you look at what's the probability of detecting the effects that you expect. So that's gray curve. You need to have the alternative hypothesis, and that's something we usually don't have, but we can sometimes estimate. And for that, we need the effect size. And the power is just 1 minus beta, where beta is the risk of not detecting the thing. So power is the probability, basically, of detecting the effect size when this effect size is real. So Cohen's d with relation with n, of course. I told you the Cohen's d is mu divided by the standard deviation of the data. So basically, your z-score is the Cohen's d divided by the standard deviation of the number of square root of the number of subjects. So what are the power issue problems? First of all, studies of low power have low probability of detecting an effect. That's clearly from the definition itself, because power is the probability of detecting the effect. And you have to think about that, because if you're investing a lot of money in a study, I mean, you have to have enough, sort of a confidence that you're going to get something out of it. Otherwise, it's a silly enterprise. Study of low power have low positive predictive value. So that's a little bit more complicated thing to explain, but I'll try. It's basically what is the probability of the alternative distribution, the alternative hypothesis to be true, when you have detected something. You may have detected something at p 0.05, even corrected, but the original probability of the alternative hypothesis is so low that this probability may be quite low. So the probability of your hypothesis, the alternative hypothesis, is not the probability that you have just from the test. You need to have what's the prior on the alternative hypothesis. If you're testing a very crazily impossible alternative hypothesis, even if you get a result, a detection result at 5%, you may have a very low predictive probability of replicating this result. That's an important thing to understand. And then study of low power are likely to show inflated effect size. And I'm going to show that in the next slides. So positive predictive value, the probability of H1 being true, knowing that you have done a detection, is something that is an easy formula to compute. It depends on the prior probability of the alternative hypothesis, and depends on the power and your alpha, type 1 error risk. So if, let's say, you have a 4 times, an odd ratio of 4 over times that H0 is true, and 1 over 5 that H1 is true, the alternative, and then if you have only 30% power, the predictive positive value would be 60%. So you only have 60% chance of replicating your result, of having actually a true result. And those are the numbers for the power. So what happens with more stringent alpha? Because in imaging genetics, we're not taking alpha 0.05, but alpha 10 to the minus 8, G-wise, if you want to correct for G-wise multiple comparison. So you see that the power is going to be very, very, very low if the effect size is not much stronger. So that's one thing that you have to remember. So effect on power goes down. Effect on positive value, it goes up, because you have a more chance that the detection is actually true. Effect on estimated effect size, the bias of the effect size goes up, and I'll show you that now. This is a simple experiment where each of these points there is just an experiment. So I'm just simulating two groups. The means are different, and I'm just going to look at when do I get a detection, and I get a detection at so the actual true effect is that red line here. But the detection is this T05 line here. So you detect here. Sorry, the true one is the dot line, and the detection when the threshold of the detection is this solid line here. So you only detect things when by chance the effect size is actually greater than the true effect size. So if you get a detection, the difference of the means that you get is always going to be higher than the actual difference, because you're detecting it, and because sometimes with noise you're getting higher, sometimes with noise you're getting lower. When you're getting lower, you don't detect it. When you're getting higher, you do, and that's the problem. So what's the mean of all those effect size when they are detected? And that mean here is that blue line here. So you see you're massively overestimating the effect size. And that's what Button showed in her paper, Nature Neuroscience Review. And you see that the lower the power, the more inflated is the effect size, which is easy to see in those formulas. So you may say, oh, that's okay, but because in our field we've got enough power, that's people don't do very, very low power study. That's absolutely not the case. We do all the time very, very low power studies, because first of all we don't know the effect size, and secondly we just don't think, okay, 50 subjects is enough. I mean, I'm talking for neuroimaging, or 20 subjects is enough. But that's not the case. Button showed that in neuroscience you got most of the studies are actually very low powered. So what is specific to imaging genetics? There's a number of things. The combination of both genetics and imaging is a problem. The combination of having to get a very large number of subjects for GWise and having them in a scan is obviously a big problem. The multiple comparison issue is very hard to deal with. The trendiness of the field, so people want to do it, and so they don't put all the means, and we get results from low power studies. And the flexibility of analysis, if you think you have flexibility of analyzing imaging data, you also have in managing imaging genetics even more. The capacity to rationalize findings, if you find a region in the brain, or if you find a gene, you'll probably find an explanation why that this SNP or this region are very reasonable to find, and that's something we is easy to rationalize. So computing effect size is really important. So for instance, I'm taking this Harry 2002 landmark paper where this was the effect size reported. So M1, 0.28, M2, the standard deviation of those two means here, and the number of subjects, 14. And what is the effect size? So you take those formulas that are easy. The effect size is just the square root of the number of subjects times the standard deviation of the means, and that would be the coherence D, and that's you get an effect size of 1.05. So that's and what's the percentage of variance explained? Well, it's not difficult to compute. I mean, please do those computations and go back to your statistical textbook and see how you do that. And that's really not a difficult thing to do. And you get that the variance explained by this SNP on the phenotype, which was the bold activity on the amygdala, is more than 40%. Is that reasonable? No, that's not reasonable. So the example, I mean, I'm taking another example of Shen using the ADNI cohort data. This is 733 subjects, and the only SNP that was found and confirmed was the APOI. And this, I'm going to show you quickly how you go from that text in the paper to how you compute the effect size. So what you do here is you take the p-value that they found, and that going from p to z, and that's the inverse survival function. That's something that you will all find in MATLAB, Python, and all those packages. And then you just divide by the square root of the number of subjects here. So this is to get to go from the z to the coherence d that I showed you before, and you get the effect size of 0.2. So effect size in imaging genetics, in general, BDNF and hippocampal volume found the homologic review found an effect size of 0.1, but Jason in his paper was finding like a percentage of variance of 0.6%, not 40%, 0.6%. That's the sort of effect size that you would probably expect. There's a good review by Flint on the effect size of intermediate phenotype that's showing that intermediate phenotypes like bold or like volumes are not much less complex than the trait of a disease, and that's something you have to remember. That they're certainly intermediate phenotype to understanding the mechanism. They are not crazily close to the gene that you would get an effect size that is very, very high with a mutation. So that's one thing that we have to remember. So COMT and DPLF, the dorsolateral prefrontal cortex, a meta-analysis showed an effect size of 0.5. That's probably well overestimated. And the HTTP, I mean, the Harry effect that I just showed you was one effect size of 1. That's a crazy number. I mean, I shouldn't say. I mean, that's a number they found, but I think it's very, very likely to be extremely overestimated. This one from Thomas was finding like a 21% of phenotypic variance and effect size of 1 as well. That's very likely to be overestimated. And the reason why it's overestimated often is that the number of subjects, again, coming back to what I explained before, the number of subjects is too small. And what you get is that early publications here, so on the x-axis is the time, on the y-axis is the effect size, and the size of this sphere, of these circles here, correspond to the number of subjects. So the bigger number of subjects. So you see that early publications have a low number of subjects and a high effect size, and then when you go down with a larger cohort, you get much smaller effect sizes, and also up to the case where you may not have an effect size at all. And that's the sort of thing that happens here as well for the BDNF, for the MIR, COMT and DPLFC paper. So what are the solutions? Well, we can start to think more about hypothesis and getting more biological knowledge to see, okay, that's my hypothesis, how much, and pre-register it, making sure that you're not going to generate a new hypothesis having seen the data, which is such an easy thing to do. We absolutely have to have an idea of what kind of effect size we expect given the literature, and that's not an easy thing to do because the effect sizes are not like prominently displayed displayed. And so you have to do what I did a little bit and go back and try to compute what is the effect size and see whether that's reasonable or not. The power analysis are completely critical. So you need to do the power analysis. You need to find out what is your likely effect size. Even if it's very uncertain, you are, having an idea is still better than having no idea. And do a power analysis with this effect size. Take robust statistical tools. I mean, we know the problems of meta-analysis is clearly the way to go. I mean, it's only when it comes to in a meta-analysis that you start to believe something strongly. And even that is not always the case. We've seen Roberto's done some meta-analysis on many low-powered studies. And all those low-powered studies were inflating the effect size and finding results that were actually not there when you looked at on a very large population. So that even those meta-analyses may be a problem. Enigma, like sharing data and sharing this, the data is really the solution to go to, where you really increase the number of subjects to make sure that the result is real. We can do better statistics and try to account from look at the effect size variation using bootstrapping, for instance. There are a number of tools, and Roberto is going to show you a couple more on how to do the effect size, the power analysis. And so but again, you have to have a reasonable estimation of what the effect size that you're hoping to have or estimating. There's a very good review from Flint et al. Recently that shows that we can actually go from randomization assignment to something called the recall by genotype, and basically choose the subjects depending on their genotype, and that would increase the power of your analysis. So I recommend that you go and see that technique. And then we need to, I think, what we're trying to do too quickly and too much in a rush. We need to get people up to the speed with the statistical tools and make sure that they know what's going on in terms of the result that they see, and that just not trust the p-value and look at more details of what are the effect sizes. And a more collaborative thing is clearly the way to go. If we can collaborate both by sharing our code and our data and making sure that we can check each other analysis and share the data such that we can get the big ends, that to me is the way to go in science, in imaging genetics. Basically trying to get more and more things that can be reproduced and you can build upon. That is, to me, the way to go. So increase the awareness of editors so that there are kind of the social solutions to the problem of reproducibility. Accept replication studies in journals. Accept pre-registration. There are a couple of journals that are starting to do that. Increase variability of analysis. So when the code and the data are available, it's much it's not easy, but it is actually possible for a reviewer to check that things seems correct. Share data and share the intermediate results. And then maybe, I don't know how to do that, but decrease the publication pressure so that we can take the time to do things with more seriousness. And that's... So there are a couple of questions that I don't know if we want to go that and if we leave that for the discussion aspect. And that Jason had for us. And I would like to thank my colleague at UC Berkeley, Mark Desposito, and Stanford, and in Saclay, and Jason, who reviewed those slides and had good comments on them. Thank you very much for your attention, and take questions. I know it's a bit, it's not always an easy message to say, okay, we need to do better.